Okay, welcome back to the afternoon sessions. Just let people come in. My name's Hannah Newbury. I am the European Medical Affairs Manager for IDEX, and it's great to see you all come back for this exciting afternoon of more lectures. Just a reminder of housekeeping. We've had lunch. So please make sure your phones have been switched off or back on silent. I'm sure you all know where the toilets are by now, but if you need a reminder, off to the left. Questions for the speaker can be submitted by the virtual attendees via chat, and we'll be keeping an eye on those. And just a reminder, these lectures are being recorded and will be made available following the symposium. I will move to introducing our speaker for this session on FGF23, What Do We Know? And our speaker will be Jonathan Elliott, who is currently Professor of Veterinary Clinical Pharmacology at the Royal Veterinary College in the UK. He's a Cambridge Vet graduate with a PhD in vascular biology. He joined the RVC in 1990 as a lecturer in pharmacology, and his research has resulted in a number of awards recognising the impact of his work on clinical practice. He's a board member of the International Renal Interest Society and was president of the European College of Veterinary Pharmacology and Toxicology. He's published more than 235 peer-reviewed original papers and reviews and supervised 37 PhD students. I will hand over to him and welcome Jonathan Elliott to the stage.
Thank you very much, Hannah. Thank you for inviting me to speak, and thanks to the sponsors of this session, Purina and IDEX. I'm going to talk about what we do know about FGF23, but obviously that will also highlight the things that we don't know. These are my declarations that are relevant to this lecture. I'm going to review the physiology initially, and then talk about pathophysiology, factors that influence FGF23, with particular relevance to the cat, wherever possible, and then talk about FGF23 in chronic kidney disease, specifically in the azotemic phase and in the early phase, and then talk a little bit about how we might use FGF23 in monitoring patients. So FGF23 was discovered in the year 2000. Like many endocrine factors, it was discovered in the diseases where it's gone wrong. And in these autosomal dominant hypophosphatemic rickets was one of the diseases that was recognized. And the genetic underpinning abnormality in that disease were mutations in 3 families with this problem that were in different parts of the FGF23 molecule. So it gave a big clue that that might have something to do with phosphate homeostasis. And those mutations led to resistance to proteolysis. And so that also gave us a clue that FGF23 is post-translationally regulated. And I'll talk a little bit more about that. And of course, then people went on to produce knockout mice and look at what happened to those mice if they didn't have FGF23. And that's what the complicated graphic on the right of this slide is showing, that by 2 weeks of age, their phosphate had gone up considerably in the homozygous knockout mice. But also their calcium goes up, and their vitamin D goes up, and eventually their parathyroid hormone secondary to the high calcium is low. And in 1997, Clotho had been knocked out and understood as an anti-aging gene. Clotho was named after the goddess of the thread of life, but it demonstrated many of the similar things to FGF23 knockout, hyperphosphatemia, soft tissue calcification, and it became clear that Clotho was involved in FGF23 action. And it is, in fact, the co-receptor. FGF23 needs to bind to Clotho as well as the FGF1C receptor in order to have many of its physiological effects. And one thing that I found fascinating in this review article that was published in 2018 is there is a relationship between serum phosphorus concentration and longevity. And here we see the Clotho knockout mouse sitting up here, and the ordinary mouse here, and we see people who live to over 100, and we see the average human. Where's it gone? Here. But if you look across species, and rodents, for example, have much higher phosphate concentrations in their plasma compared to our species, they live a shorter period of time, and there is that relationship. And actually many of the abnormalities in an animal that's not able to regulate its phosphate are premature aging effects. Decline in circulating Clotho obviously causes that premature aging. And Clotho is a major regulator of antioxidative stress pathways. So the NRF2 pathway is the major antioxidative pathway. Regulatory, it's a transcription regulator of a number of different genes. So that raises the question, which I'll revisit in the diet talk as well, about whether chronic kidney disease accelerates the aging process. But let's return to phosphate homeostasis. We're talking about the balance between intake and excretion of phosphate on a daily basis when the animal isn't growing, it's not lactating, it's not pregnant, and intake needs to equal excretion. And it's the intestines, the bone, and the kidney that play a major role in maintaining that balance. And FGF23, alpha-Clotho, PTH, and calcitriol are the hormones that are there to help that. Now, before 2000, we thought that PTH and calcitriol were doing much of the regulation, but we couldn't really understand that because they were regulated by calcium. And I think it's a bit simplistic to think that FGF23 is regulated by phosphate, because calcium and phosphate need to exist together, and they're close to their limit of solubility in mammalian fluids. And that's necessary in order to form bone so that you can walk on land. So that transition from water to land, evolutionary, there needed to be a way in which you could have calcium and phosphate at high enough concentrations to form bone, but some way of preventing them calcifying the tissues that you didn't want to be calcified. And so these systems have evolved, and I think Dottie mentioned calciprotein particles in passing in her talk. Calciprotein particles are a colloidal form of carrying calcium and phosphate together in a suspension such that they don't precipitate out in the soft tissues. And these calciprotein particles form by binding of complexes of calcium and phosphate with a protein called fectuin A, and those monomers then form into heteromers, and the particles grow. And it's mainly the calciprotein monomers and the early particles that are physiologically important for chaperoning calcium and phosphate around the body to areas where we want bone to form and to carry it away. And there are scavenging receptors in the liver to remove that. Those larger particles, and the kidney probably excretes the calciprotein monomers. And we'll return to that soon. So that understanding of these particles that are nanoparticles, they're only a maximum of 250 nanometers in diameter, and those are the pathological ones that actually cause inflammation and probably lead to or are important in vascular calcification. So how is FGF23 controlled? What are the mechanisms? And this is an area that we don't know for sure what's actually happening, but we do know that it's probably not stored in secretory granules like many hormones, but it's continually made but degraded, and the degradation process is a proteolysis. And there are enzymes that will protect it from proteolysis. So if it's O-glycosylated by this enzyme GLANT3, then it's much more slowly destroyed by proteolysis. And that explains why there's a delay between a signal such as an increase in serum phosphorus concentration and secretion of FGF23, and that delay can be around about 8 to 24 hours actually. And the factors sensing phosphate have been an area of active research, and there are 4 factors that have been proposed that I'm aware of: the FGF1 receptor that binds phosphate, calciprotein particles themselves, phosphate transporters, and most recently glycerol 3-phosphate released from proximal tubular cells, giving a communication line between the kidney and bone. All of those proof of concept of those can be shown in cultured bone cells, but whether that's relevant to the physiological effect of phosphate on FGF23 secretion is really not well established. But what we do know is that calcium is needed for FGF23 to be secreted. You need to have an above threshold ionized calcium for FGF23 to be produced. And that makes absolute sense, because what FGF23 is doing is inhibiting phosphate entry into the body, but you don't want it to do that when the hormones that are raising calcium are bringing phosphate in. So if you need the ionized calcium to go up first, and then you need to prevent the phosphate coming in and overshooting and causing that soft tissue mineralization. So I feel that this system has evolved to allow calcium regulation at a fine level without causing phosphate to overshoot and lead to soft tissue mineralization. And this diagram just illustrates that. So it is too simplistic to think that it is just phosphate that's regulating this hormone. And we know that FGF23 inhibits PTH secretion, it inhibits vitamin D formation in its active form, causing more 24-25 dihydroxycholecalciferol to be made. It increases phosphate excretion, and therefore, and via vitamin D, reduces phosphate absorption. So it acts as a brake on phosphate rising in response to PTH and calcitriol once calcium is normal. And all the physiological actions of FGF23 are require the cofactor klotho. Indeed, klotho itself is released from the kidney, where it's expressed most highly, and has effects that are independent of FGF23 that are related to calcium homeostasis. So what do we know about the cat? Well, some of this overlaps with what Dottie was saying in her talk. There's no change in FGF23 when you give a meal that contains readily available phosphate as an acute feeding study. And this is one of the studies that was done by the Waltham Pet Science Institute, feeding a diet that had added sodium dihydrogen phosphate, and that's rapidly absorbed, as shown in the top panel here, giving a nice rise in serum phosphorus. Whereas if you feed a diet that has no inorganic phosphate that's readily available, then you don't see that postprandial rise in phosphate. In fact, you see a slight fall in phosphate as insulin secretion drives phosphate into cells. Associated with that rise in serum phosphorus is a fall in ionized calcium and an increase in PTH, but no change in FGF23 acutely. But if you feed those diets longer term, then FGF23 certainly increases. So FGF23 is responding in a longer term process. And that's the chronic feeding studies that were done by the Waltham Centre. All of those data you can model to look at what are the major factors controlling serum phosphorus. And that path analysis was done by one of my PhD students, because there wasn't anything she could do prospectively during COVID. So these data were really useful, and we're preparing a paper based on that path analysis showing that actually FGF23 and urine excretion of phosphate is the major factor regulating serum phosphorus when you're feeding a high level of inorganic phosphate. Interestingly, if you have no inorganic phosphate in your meal, you need to remove phosphate from bone in order to maintain homeostasis. So I don't think a diet that has no inorganic phosphate there is a natural diet that will have no consequences on the animal's physiology long term. The calciprotein particles definitely have an influence on FGF23 secretion, and these data have been published very recently showing a postprandial rise in calciprotein monomers in people, and that rise is higher in people with chronic kidney disease. And actually the same group have very recently published a paper showing that that rise in calciprotein monomers is a risk factor for progression of chronic kidney disease. We have very limited data on cats with calciprotein particles, and Pat Can Tang will be presenting a talk at this meeting on his work on this, where we have demonstrated that high-density calciprotein particles are higher postprandially. These are in cats that have got chronic kidney disease, and that there are relationships with phosphate, calcium, and FGF23 with these calciprotein particles. So this physiology and pathophysiology definitely applies to the cat. Returning to the feeding studies that the Waltham Pet Science Institute did, where they looked at different levels and different calcium to phosphorus ratios in diets that contained 1 gram per megacal of sodium tripolyphosphate. There were 3 different diets. Control had no added sodium tripolyphosphate. The moderate diet had 1 gram and 4 grams total phosphate, with a calcium to phosphorus ratio of 1.04, and the high diet had 5 grams total, still 1 gram of STPP, but a calcium to phosphorus ratio of 1.27, showing how important that calcium phosphorus ratio is on bioavailability of phosphate, because the moderate diet showed the significant increase in FGF23. But notice that increase very rarely went above the 300 picograms per mil, and by the time those cats had been on that diet for a couple of months, all of the values were below the 400 picograms per mil in that diet. Now, these cats were followed for 6 months, which actually is a significant proportion of their life, and there was no evidence of any structural changes in the kidney by ultrasound. There was no change in urine protein to creatinine ratio, and there was no difference in glomerular filtration rate in these cats. So, and bone structure was also examined. So to all intents and purposes, these diets had no observable effect on the cat. So I would be confident that 1 gram per megacal of sodium tripolyphosphate added to a diet would be safe to feed. Obviously, the other components in the diet could affect the bioavailability. So one thing that I think is obvious about nutrition is it's really complicated, and if you change one factor, that isn't the only factor that changes about the diet. So what are some of the pathophysiological factors that influence FGF23? Well, Natalie Finch in her PhD demonstrated very nicely that there was a relationship between FGF23 concentration and GFR. So the intact molecule is renally cleared, and you need to take that into account when you're interpreting FGF23 levels. Increased calcium and phosphate are important. So Rebecca Geddes in her work demonstrated that cats with chronic kidney disease in stage 3, for example, as shown here, if they had phosphate within the iris target range, their FGF23 levels were lower significantly, and this is a log scale, so it really is highly significant, compared to those cats that had serum phosphorus above the iris target level. Inflammation and, she also went on to demonstrate that total calcium was an independent factor regulating FGF23 or associated with FGF23. And the relationship that she found between these multiple factors and serum FGF23 concentration had an R-squared value of 0.78. So we were looking at most of the factors that are influencing FGF23 concentration. Outside of the cat, we know that inflammation is important and can regulate FGF23 concentration. So in the later stages of chronic kidney disease, where there are pro-inflammatory factors ongoing systemically, then the IL-6 and STAT3 pathway is probably regulating FGF23 and upregulating it. With iron deficiency, FGF23 is upregulated, and EPO has effects as well. And the uremic toxins are also important in influencing FGF23 concentration in later stages of kidney disease, and workers from Taiwan demonstrated that in cats very nicely. We also studied magnesium. Magnesium is a bit ignored in small animal veterinary practice, and it shouldn't be. And hypomagnesemia makes mineral bone disorder worse. At every stage, iris stage of CKD, the hypomagnesemic cats had higher FGF23 concentrations compared to the normomagnesemic cats. And that proved to be statistically significant overall, that magnesium is influencing FGF23 secretion. It's really complicated, the effect that iron deficiency has on FGF23 gene expression. It's upregulated by both iron deficiency and anemia. And that interacts with inflammation, because actually inflammation exacerbates iron deficiency, with hepcidin being produced and inhibiting iron absorption from the gastrointestinal tract and iron release from cells that store the iron following degradation of red blood cells. So FGF23 is upregulated by inflammation and by uremic toxins. Interestingly, one of the most effective ways of reducing serum phosphate and FGF23 is to use an iron-containing phosphate binder. And that's been shown experimentally, but it's also been shown in meta-analysis studies in human patients with chronic kidney disease. So actually, the iron factor may be more important than we think in CKD. So let's move on to azotemic chronic kidney disease, or the different stages of chronic kidney disease. What's accepted in the human field is that FGF23 is the first thing to increase in kidney disease. And we're seeing 40% of these human patients, this is obviously a very large study, which have an estimated glomerular filtration rate of greater than 70, which is actually approaching normal. So these are really quite early CKD patients. 40% of those had above reference interval FGF23, and it then carried on increasing. That doesn't give you an idea of the magnitude of the elevation of FGF23, which is exponentially rising as the kidney disease gets worse. But it tells you that it goes up first, and then PTH goes up, and then calcitriol comes down, and eventually phosphate in the serum goes up. So looking just at phosphate in the serum doesn't really tell you how this system is dysregulated at an early stage. Does that apply to the cat? Well, we see this exponential rise with stage of FGF23. Again, a log scale. This is work that Rebecca Geddes published in 2013 in cats. Group 1 are actually normal healthy cats, and Group 2 are azotemic CKD cats in stage 2, and Group 3 stage 3, and Group 4 stage 4. What we do know is that one of the reasons why FGF23 becomes completely dysregulated as the stage increases is because of clotho deficiency. And that seems to be the case in human medicine. We haven't been able to measure serum clotho in a biologically sensible assay for the cat, in an assay that makes biological sense, I should say. But we have been able to measure expression of clotho in kidney obtained at post-mortem from cats that were post-mortem because they were euthanized at different stages of chronic kidney disease. And we've correlated Klotho expression with the pathology in the kidney, and it correlates pretty well. So the more severe the pathology in the kidney, the lower the Klotho expression. And FGF23, I'll show you data in the cat, seems to predict progressive chronic kidney disease. So its increase heralds poor prognosis, in other words. That begs the question whether interventions that influence FGF23 influence progression and survival in cats. Is it just a marker, or is it a driver of that progression? It could be both, actually. Again, this is work that Natalie Finch did in her PhD, looking at the early stages of chronic kidney disease. And to my mind, really the best identification of an early stage kidney patient is prospectively following cats and identifying subsequently that they have chronic kidney disease that we all know and love. And that's what this study was. It's screening old cats, following them for 12 months, and then categorizing them at the 12-month time point into they've got azotemic chronic kidney disease, they're completely healthy, or they're borderline. And that's what group 1 is, completely healthy. Group 3 is they've got azotemic chronic kidney disease, and group 2 is they are borderline. And you can see that the FGF23 values measured at screening, that's 12 months before they had developed the azotemic chronic kidney disease, were significantly higher in that group 3 compared to group 1. They were also significantly higher in group 2 compared to group 1. Whereas their plasma phosphates and their plasma creatinine concentrations at the time of screening were not different at all between the groups. We then went on and looked at SDMA as an early indicator of chronic kidney disease. And we did this retrospectively, again during the pandemic, looking at samples that had been obtained from our archive where SDMA was elevated, and compared them to samples where SDMA was normal. So SDMA above 14 or above. And we looked at the proportion of cats that had FGF23 above 300 picograms per mil, and you can see that there's a threefold difference between those that had elevated SDMA and those that had normal SDMA, 21% and 62%. And if you make the cutoff higher, then you obviously pick out fewer cats with elevated FGF23, but you differentiate more clearly between the two groups. And so this is one of the reasons why we picked 400 picograms per mil as being a threshold to suggest these cats need phosphate restriction. So just to conclude this section, a reduced glomerular filtration rate or an elevated SDMA or a creatinine increasing within the reference range, provided those are persistent, gives you an indication that there may be early stage chronic kidney disease going on. But if there's no evidence of active disease, what do we do to treat that? We continue to monitor at the moment. If there is evidence of active disease, and we're getting better at identifying markers of active disease, then there may be something that informs you about how to treat that. And we heard about cystatin B this morning. Urine protein to creatinine ratio may be elevated, and that would give you a reason to direct your therapy at proteinuria. But if FGF23 is elevated, then it gives you an indication that perhaps dietary phosphate restriction is necessary, which we'll talk about in the next talk. And certainly if that's greater than 400 picograms per mil, then we would recommend at the moment that dietary phosphate restriction is indicated. What about the azotemic cat? When we see azotemic cats, some of them are hyperphosphatemic for their stage and some of them are not. And if you phosphate restrict them, those that are hyperphosphatemic for their stage show a nice reduction in their serum phosphorus. So there's no problem with monitoring those cats in response to their dietary treatment. And the same is true of PTH. The PTH goes down, so the dark purple are the cats that are hyperphosphatemic for the stage, the lilac or the light purple are the cats that are normophosphatemic for the stage. No change in the normophosphatemic in terms of their PTH, so it's hard to monitor what's going on in those cats. Whereas if we look at FGF23, and again it's a log scale here, we see a reduction in FGF23 in the cats that are treated that are hyperphosphatemic, but we also see a similar reduction in the FGF23 in the cats that are normophosphatemic for their stage, suggesting that that phosphate restriction is depleting whole body phosphate stores and in some way helping with this bone mineral disorder. And the percentage reduction, a little bit lower in the group that were normophosphatemic for their stage, but nevertheless, a significant reduction, and a reduction that human medicine would actually be really pleased to be able to achieve, because compliance with diets and so on in human patients is really poor, and they need often to use phosphate binders. So just taking from that study some individual cases just to illustrate the point. Here's a case in Irish stage 2 CKD. The plasma phosphate at diagnosis was well within the reference interval, yet the FGF23 at diagnosis was 1,232 picograms per mil. When we saw the cat after 5 weeks of phosphate-restricted diet, the plasma phosphate was 1.01, so no real change there. But the FGF23 had come down to 561 picograms per mil, and that's quite acceptable to us. Anything below 700, I think, in this stage of chronic kidney disease is doing well. Here's a hyperphosphatemic cat, so we probably wouldn't need to measure FGF23 at diagnosis of CKD. The cat had a phosphate of 2.38 millimoles per litre. But we did, because we're doing this as research, and it was 1,500 picograms per mil. After 6 weeks of phosphate restriction, we had the phosphate within the target range. But the FGF23 was 1,246. Now it's come down a bit. If it continues to come down, then that is good news. The cat is responding to the treatment. It may need further phosphate restriction in order to reduce the FGF23, so phosphate binders might be needed. The question would be, is this cat hypercalcemic? You need to check the calcium in this cat, because FGF23 will be higher than it should be if the calcium is elevated. So continuing to monitor, that will be the emphasis on my diet study, on my diet talk, is that monitoring is the thing that most practitioners don't do enough of in order to know whether the diet they've prescribed is actually the right one for the cat. Rebecca Geddes published these data on survival, where FGF23 was an independent factor at diagnosis predicting survival. It was a poor an indicator of poor prognosis, independent of age, creatinine, and urine protein to creatinine ratio. And these are the data split into quartiles. The lowest quartile had less than 700 picograms per mil, and that's a reasonable target at this stage. We don't really know what the target is for each stage. Not only that, but in that study, Rebecca also looked at progression, classification of progression based on a 25% increase in creatinine within the first 12 months from diagnosis. And she was able to look at that in 214 cats where we had retrospectively enough plasma in the freezer to measure FGF23. 109 of those had short follow-up, so we weren't able to classify them as progressors or not, but we had 38 progressors and 67 non-progressors over that 12-month period. And FGF23 was the only factor other than age that stayed in the multivariate variable analysis as an independent predictor of progression. And that meant it kicked out things like packed cell volume, urine protein to creatinine ratio, and serum phosphorus, which in our previous studies had been significant predictors of progression. So why is it that FGF23 is such a good predictor of progression? We've carried on looking at progression in more cats. Over the last 10 years, we've seen more cats. We're looking at the rate of change of creatinine from diagnosis, because we recognize that cats progress at different rates. And we see linear progression, where the creatinine gradually increases over time, and we see what we call stepwise or non-linear progression, where all of a sudden the creatinine increases. And generally that happens within the first year of diagnosis. I think the writing on the slide on the graphic on the right will not be visible, but essentially that is showing that in the 2 years following diagnosis, the non-progressive cats generally remained non-progressive. Whereas obviously if we've classified them as progressive within the first year, they were progressive. But if we look at the changes in creatinine over time, and we hive off the top quartile, which are the fastest progressors, based on their slope of creatinine against time, you could make that as 1 over creatinine against time, as the fast progressors, and look at them compared to the rest, then we actually demonstrate in this slide that those fast progressors have significantly higher FGF23 concentrations at diagnosis compared to the slower progressors who have lower FGF23. They are quite a lot of overlap between the two groups, as you might imagine. If you just look at progressors versus non-progressors, the non-progressors have even lower FGF23 at diagnosis, and are significantly different from the progressors. Again, this is a log scale, so the differences are bigger than they appear, but there is a significant overlap, and what we need is more factors that help us to distinguish between progressors and non-progressors and fast progressors, because these are the ones that really need very intensive monitoring. So why should FGF23 be associated with such poor outcome? Well, the theory as to why phosphate reaching the later parts of the proximal tubule, the S3 segment, can cause damage is that these calciprotein particles bind to TLR4 receptors, become internalized, and get stuck in the lysosomes and cause oxidative stress. Experimentally, increasing FGF23 indicates loss of clotho, and clotho is one of the major defense mechanisms. Increasing FGF23 indicates that phosphate is not being reabsorbed in the early part of the proximal tubule, so its concentration will go up. But once FGF23 is going on that exponential rise, it then starts to have clotho-independent effects on the cardiovascular system, so hypertension and cardiac hypertrophy. And FGF23 is influenced by the later complications, such as iron deficiency and anemia and systemic inflammation, so the later complications of chronic kidney disease. And so it's a kind of positive feedback cycle as to why FGF23 elevation is going to give rise to poor outcomes. And that's certainly the case in human medicine, and even better if you can do a ratio between FGF23 and clotho, because clotho goes down, FGF23 goes up, and in the the lowest quartile of that ratio, adverse renal events were much more common and occurred at a much more rapid rate in this large human study. So, mineral bone disorder in our species is an important consequence of reduced renal mass, and it's initially an adaptive response, but it soon becomes maladaptive, and it probably is already maladaptive by the time we diagnose chronic kidney disease. In the later stages, it heralds progressive kidney disease and all-cause mortality. It predicts those. It can be managed by dietary phosphate restriction, which reduces azotemic crises and all-cause mortality, and I'll show data in the next talk about that. FGF23, I hope I've demonstrated, has utility in monitoring response to treatment and personalizing that treatment, and identifying cats that really need to be monitored and followed very closely compared to those that have stable kidney disease and can be monitored less closely. Thank you very much.
And does anybody have any questions for Jonathan? Very comprehensive. No, then in the interest of time, I think we'll move straight on to the next talk. Did you want to?
Yep. Yep. So just then let's go to the next talk that will be about the selection of the diet, right? So just a reminder for everyone that the next break will be at 3:50 until 4:20 and will be after this lecture. Okay?
Okay. So I just need the slides.
They are on it, they are on it. So I take the opportunity then for introduce myself, that I have not done it. Meanwhile, they put the,
Here we
the
go.
slides. Okay. Then afterwards.
So I declare that I'm a pharmacologist, not a nutritionist. So I'll get that out there up front. And I also declare that nutrition studies are really difficult to do, and particularly those around kidney disease, early kidney disease, in terms of characterizing the patients that you enter, making sure the groups are balanced, and carrying them out for long enough in order to determine whether there is an effect. That said, I have the same disclosures as before. I could have done this talk very quickly. I feed Royal Canin diets. They fund the research that I do. But I decided to do a little bit more than that. Okay. So I'm going to talk about dietary treatment of the azotemic cat, because that's what we know most about. And I think that's what most of the diets are based on. I'm going to talk about the composition of the standard renal diet, recognizing that there is no standard, but there are some features that are common across the diets. I'm going to emphasize, as I did in the previous talk, that monitoring and adjusting treatment is really important. And I'll talk a little bit about the controversy that Dottie mentioned about protein levels. And then I'll talk about what we don't know about the non-azotemic cat that we think has chronic kidney disease. I've just put up 6 diets that are from 3 different companies, which you'll probably recognize as probably the major players in the clinical nutrition field, just to say they have lower protein than a standard maintenance diet, which might have around about 100 grams of protein per megacal. They have enriched N3 polyunsaturated fatty acids at around about 2 grams per megacal, and they have lower phosphate than a standard maintenance diet. Now, Dottie showed some figures. Standard maintenance diets vary considerably with the amount of total phosphorus, but these are generally less than 1 gram per megacal, which is significantly below the 3 grams per megacal, which is the median value for the maintenance diets. They're also lower in sodium, and sodium also varies quite a bit between different diets, but generally these are 50% lower than, so they might be 1.2 to 2 grams per megacal in a standard maintenance diet. So I think there's strong evidence, and some of what I'll talk about will repeat what Dottie said this morning, or this lunchtime, for phosphate restriction. The protein needs to be of high quality, easily digestible, and it's always above 50 grams per megacal in the renal diets. That's controversial. There is no evidence that restricting protein slows progression in experimental models of kidney disease in the cat. However, it's quite difficult if you're using animal protein to produce a diet that's low in phosphate without reducing the protein somewhat, because many of the protein sources are also sources of phosphorus, as I understand it. There is some clinical evidence that N3-PUFA supplementation is beneficial, and that's from an epidemiological study, a retrospective one, where the diet that prolonged survival the most had 1.96 grams per megacal of N3-PUFA supplementation, and there's experimental evidence from other species. Sodium reduction doesn't really assist in blood pressure control in the cat, and activates the RAS system. Is that going to be beneficial? Probably not. The antioxidants are all provided. It's difficult to quantify how much antioxidant potential a diet has. Vitamin E and vitamin C and so on are fortified in these diets, and it's poorly studied in cats. I think it's an area that we need to do more research on. So these are the data that Dottie mentioned previously from Linda Ross in the experimental model. I would suggest that the normal phosphate diet was actually a high phosphate diet, and you don't need statistics to see the difference in the pathology of the remnant kidney after a year of allowing this model to progress or not. And it didn't progress functionally, but it did progress pathologically. Our study was a randomized, but it was controlled in that we had a group of cats that wouldn't eat the diet we offered them. The diets in those days were not very palatable. They were little like paste that stuck to the roof of the cat's mouth. The introduction of the dried version actually was a big advance, and I think we're really fortunate today that there are multiple diets on the market that have those features that reduce phosphate intake, that supplement N3-PUFAs, and so on. But the difference in survival here was quite significant, 633 days versus 264 between the two groups. And we were trying to control phosphate and PTH in the group that were receiving the renal diet. We decided not to give phosphate binders to the cats that were not able to be transitioned onto that renal diet. What we also found was that more of the cats, or a higher proportion of the cats that were on their maintenance diet, died of chronic kidney disease, whereas more of the cats that were on the clinical renal diet succumbed to cardiovascular disease. Now, those are just observations. They're not statistical. The study was not powered sufficiently to really look at the cause of death in those cats. But I think it's intriguing. One of the things that's said is that you don't really need to start clinical renal diets until stage 3. If you divide the cats in our study into stage 2 and stage 3 at diagnosis, these are the stage 2 cats. The difference, the purple line is the diet-fed cats. The green line is the cats on their maintenance diet. There's still a significant difference, even though the group sizes are much smaller. In stage 3, the difference is similar in magnitude. KD did just the same, although KD was done on a randomized controlled study, and the control diet was standard between all of the cats. It would have taken us 10 years to do that, because our cats that didn't eat the diet were used in the study. Obviously, the control diet in this study was a better quality diet than the cats were already on, because they didn't see so much mortality in their study. But both renal survival and and uremic crises were significantly different between the two groups. And of course, we can't conclude what factor in the diet is causing that difference. And there are multiple factors that could possibly do that. But KD at the time had 1.1 gram per megacal of phosphate, and the control diet had twice that amount. It's often asked whether it's possible to use a standard maintenance diet and just add phosphate binders. And this is the only data that I'm aware of that's available to address that question, which was work done by Scott Brown and his group at Georgia in nephrectomized cats, so a surgical mass reduction. These were cats that were in stage 2 or lower, non-azotemic stage 2 or lower after 6 months following the 11/12 nephrectomy. The base diet had 4.36 grams per megacal of phosphorus, so a reasonably high level of total phosphorus, 4 times the amount that's in standard renal diets. This was a randomized crossover study. They were fed for 56 days. There were 10 cats involved. And what you can see is that the serum phosphorus, when they were on the diet plus the binder, was significantly lower, although didn't meet the IRIS target range as the mean value. Serum PTH and urinary excretion, fractional excretion of phosphate was also significantly lower. And they took 6 cats and carried them on for 9 months. And they did meet the IRIS target range after 6 and 9 months on this regimen. So it is feasible in the early stages of CKD in normal healthy cats that have had surgical reduction in their renal mass to control phosphate. Whether that's the case in older cats with naturally occurring chronic kidney disease, we don't have any evidence for that. So the question about protein in these diets, is the level of protein in the diets detrimental to the cats? There has been no head-to-head study in azotemic cats in stage 2 or stage 3 using renal diets of different protein content. And I think the source of protein is also important, and the amino acids within the total amino acid availability is also important. There are some data from the human field suggesting that non-diabetic kidney disease patients of a similar stage, the staging system is different in people, do benefit from protein restriction, time to dialysis or renal death. And this is a Cochrane meta-analysis of 1,500 patients. But there are multiple factors that contribute to uremic sarcopenia. So the argument that lean body mass reduction is enhanced by restricting protein, well, inflammatory cytokines, angiotensin II, metabolic acidosis, growth hormone, IGF-1 and PTH, inactivity, obviously in people, cats are always inactive, and arthropathy may well influence sarcopenia. And we really haven't studied sarcopenia in any depth at all in cats. And low protein and low energy intake can give rise to that protein energy wasting. But what we do know is that if you feed higher protein, then you get more generation of uremic toxins. And these are data from the Hills group looking at higher protein diets and uremic toxins in the colony cats that had some reason to decide that they were in stage 1 CKD. And there are 3 diets fed here, 37, 32, and 26% protein. And if you just look at endoxyl sulfate, you'll see that the levels, the relative units, these are mass spec measurements, were significantly different and proportionate to the level of protein in the diet. So there is a reason why protein restriction could be beneficial in terms of uremic toxins. And we do know that endoxyl sulfate is related to FGF23, and it has an influence on, in the later stages, on survival. It's a gut-derived uremic toxin. Its production is following the metabolism of tryptophan in the by the colonic bacteria, and then that's converted into endoxyl sulfate by the liver in the animal itself. And you can see this data from Jessica Quimby's group, showing that indoxyl sulfate is higher according to the stage, and that intestinal diversity of bacteria is lower. And if in another paper that they published, they also demonstrated that fecal branch-chain fatty acids were higher in those cats with chronic kidney disease that had muscle atrophy. And the fecal branch-chain fatty acids are the detrimental fatty acids produced by the bacteria that generate the uremic toxins. And as you went through the different stages, so you saw an increase in muscle wasting. And so another study that was published by the Hills Group in Metabolites in 2019 showed that feeding functional food diets with enhanced antioxidants, increased fiber, sources of protein from plant sources, vegetable sources, and so on, had an effect in aged colony cats of reducing indoxyl sulfate, increasing glomerular filtration rate, and reducing SDMA. So that there may well be an effect of diet on uremic toxin generation, which actually talks to the quality of the diet and the quality of the protein that's in there. Just a few words about metabolic acidosis. As Dottie mentioned this lunchtime, this has not been well studied, and the dietary management of acidosis has not been well studied in the cat. We do know that acidemia is not seen until stage 4 CKD in cats in general, but are cats buffering excess acid that's not excreted by the kidney in bone? That's a possibility. Professor Syme did a study in her PhD looking at potassium gluconate supplementation. The hypothesis was that this might lower blood pressure, but obviously gluconate is also an alkalinizing agent. And it was a randomized controlled study, one of the first ones that we did. Unfortunately, we didn't get the color of the powder the same between the placebo and the control. So we learned that that's important, because owners tell you what color capsule they're giving, they're sprinkling onto their cat food. It was 12 weeks in duration for each arm, and we definitely got the potassium gluconate into the cats because the urine was alkalinized and the potassium went up. But we had no effect on body weight over that 12-week period, no effect on blood pressure, which was what we were looking for, and no effect on markers of bone turnover. So providing alkali to base diet doesn't seem to influence bone turnover or body weight in that 12-week period in cats that were fed, actually those were the Waltham diets at the time, and therefore I think we do need to do more work in this area, and it's good to see that people at UC Davis are actually taking more interest in metabolic acidosis in cats with chronic kidney disease. I think this paper is really interesting and illuminative, and that's Kathy Freeman's work looking at growth curves in cats that have kidney disease from 6 veterinary practices. And they were able to look at them from the diagnosis, but actually retrospectively going back before the diagnosis. And the different colored lines, blue, red, green, and purple, are increasing age of the cat. So the cats were losing weight and before they were diagnosed with chronic kidney disease. The older cat group were losing weight more rapidly. And obviously you can see that the loss of weight increases as kidney disease is diagnosed. But these cats were losing 8.9% on average of their body weight in the 12 months before diagnosis. And they were being fed standard maintenance diets. The survival time in cats weighing less than 4.2 kilograms was shorter than the survival time of cats weighing more than 4.2 kilograms, which I guess is something that we recognize, that the heavier cats live for longer. Does that mean that measures that maintain lean body mass would improve survival and slow progression? Would feeding more protein be counterproductive if reduced protein intake is not the reason for the loss of lean body mass? those are questions that we don't know the answers to, I think. What we do know is that there's an increased risk of hypercalcemia developing when you feed a phosphate-restricted diet to a cat with chronic kidney disease. We don't know whether it's definitely feeding the diet that's causing the hypercalcemia, because we see hypercalcemia in cats that are not on renal diets as well, and we see them at diagnosis of chronic kidney disease. But as we follow them over time, this is work from Henk van der Broek's PhD, we see that the incidence of hypercalcemia is much higher in cats with chronic kidney disease compared to cats that are healthy and that we're monitoring over time. Transferring cats in the group that develop hypercalcemia to a less phosphate-restricted diet, as was mentioned earlier, seems to help the hypercalcemia to resolve in many cases. And our current strategy is to transfer these cats from the renal diet onto the early renal diet. And these are the data that Dottie referred to. These are the cats that developed hypercalcemia when they were transitioned onto a diet. They're shown in the dotted lines, and you can see that their ionized calcium goes down and their PTH comes back up into a measurable range when you transfer them onto the less phosphate-restricted diet. But the cats that were hypercalcemic at diagnosis are the solid line on those two graphs, and they don't change in terms of their calcium value. So it's not a treatment that we find works for cats that are already hypercalcemic before they've been transitioned onto the diet. So that was the azotemic cat. Now, what do we know about the non-azotemic cat in stage 1 and non-azotemic stage 2 cats? And again, I could make this very short, very little, because there have been no studies in naturally occurring cats that are owned in the community. There have been some studies that have demonstrated an effect of dietary management, in my view. So there's little evidence to base this on. But what we do know is that there are diets that are designed for early kidney disease, and they have more protein in them than the diets that are designed for azotemic kidney disease, and they have slightly higher phosphate. Some of them have the same level of phosphate as the standard renal diets. I'm going to talk about two studies. One was published in PLOS One in 2016. It was a prospective study involving cats recruited through 11 veterinary practices in the US. It was conducted by scientists at Hills. The test diet was Hill's protective diet, fortified with carnitine and amino acids and antioxidants. And it had 2.2 grams per megacal of phosphorus. One of the things about the dietary literature is you have to convert everything into a standard, because they keep putting percentage dry matter and all of this. Anyway, that was my grouse. It had 95.4 grams per megacal of protein from various sources, including different vegetable sources. And it was, as I said, mentioned, it was fortified with carnitine. Cats had to be indoor cats. They had to be 9 years or older without significant medical condition. And the main data from this study, which was a 6-month study, is shown in the table here, where creatinine reduced in the cats on the test diet, and SDMA didn't change, but SDMA increased in the cats that were on their standard maintenance diet, and their creatinine values didn't change. So the conclusions that were drawn from this study is that cats fed owner choice food were more likely to have progressive renal insufficiency. And that was based actually on sub-analyses of some of the cats that had an SDMA of greater than 14. My view of this is that that conclusion seems to be too strong based for the evidence that was presented. Really, have they demonstrated progression in that study? The endpoint wasn't designed before as the study was designed, and I don't think the study was long enough. But it's difficult to do these studies. It is really difficult to do these studies. I showed these data in the last talk. But these data stimulated us to do a prospective randomized controlled trial looking at phosphate restriction in normal healthy cats, a proportion of whom we expected to develop azotemic chronic kidney disease over the period of the study. So this was a randomized blinded controlled clinical trial. We had a goal to recruit 30 per group, and it was powered assuming that 33% of the cats fed the maintenance diet versus 5% of the cats fed the test diet would develop azotemic chronic kidney disease in 18 months, based on our previous data. And the end point was a persistent serum creatinine above 2 and with a urine-specific gravity of less than 1035. That was a progression to azotemic kidney disease. The test diet had a protein content of 76 grams per megacal and a phosphate of 1.6 grams per megacal. And it was the Royal Canin early renal diet, and they funded the study. The control diet had 86 grams per megacal of protein and 2.6 grams per megacal of phosphate, and was a good quality maintenance diet. In the interest of time, we screened 145 cats, excluded 73, many of which had hypothyroidism or chronic kidney disease, unbeknownst to their owners, and 72 were randomized, 37 to the test group and 35 to the control group. We analyzed data from 26 cats fed the test diet and 28 cats fed the control diet that completed the 18-month study. The exclusions are shown in the consort diagram in the paper. Plasma phosphate and log FGF23 concentration didn't change over time. In a sub-analysis of those cats that at entry had an FGF23 of above 150 picograms per mil, we did demonstrate a reduction in their FGF23 on the test diet. The body weight and the body condition score did decrease, and the muscle condition score did decrease over time. So body weight is shown here, and it's very similar to the data that was shown in the Freeman study, with around about less than 10% change in the body mass over a 12-month period. A body condition score similarly reduced, but it wasn't diet related, it was happening in all of the cats. Total calcium and ionized calcium increased in both groups over time, therefore seems to be an aging effect, and interestingly total T4 also increased significantly over time. Plasma creatinine and urine-specific gravity decreased over time. We did this study before we had SDMA available. That might have been an interesting thing to measure alongside creatinine, because presumably the creatinine reduced because the muscle mass reduced in these cats. These effects appear to be aging effects. 3 cats per group developed persistent azotemia, so the prevalence was 11% across the whole population, and disappointingly not enough for us to really show any effect. Fractional excretion of phosphate changed significantly over time in both groups, and it reduced in both groups, suggesting that our control diet had less phosphate in it than on average the cats were being fed before entering the diet. And after 15 months of the study, the two groups diverged from each other in terms of fractional excretion of phosphate. So there was a difference between, and an effect, therefore, of the test diet having a lower fractional excretion of phosphate compared to the control diet. But it took some time for that to become evident. And PTH was lower and was less likely to increase in the cats that were fed the test diet. And PTH was actually, because of the increase in calcium, PTH was actually quite low in some cats. So we analyze this as a categorical variable, because obviously a PTH below the reference interval, we can't attribute a value to it. 6 cats developed ionized hypercalcemia. 5 of them were in the test group and 1 was in the control group. Now that wasn't statistically significantly different, but it clearly indicates that a more restricted phosphate diet is probably associated with the hypercalcemia. And PTH was lower, therefore, and there was a reduced risk of PTH increasing in the cats that were fed the test diet. So what could we conclude from that? Well, we didn't have enough cats developing azotemic kidney disease for us to really conclude anything. There was no demonstrable effect of feeding an early renal diet on predefined endpoints. The prevalence of azotemia was lower than we expected. Loss of body mass and body condition score led to a reduction in plasma creatinine concentration. Both test and control diet were probably of better quality than the cats were eating before they were transitioned onto that diet. And Dottie mentioned these studies previously, but this is what was called the FOS2 study, where the test diet contained just under 3.6 grams of phosphate in total per megacal, 1.5 grams of which was sodium dihydrogen phosphate. And there was no change over time, over a 6-month period, 28 weeks, that these cats were fed this diet, but the urine albumin to creatinine ratio went up, and 9 out of the 25 cats in the test diet group eating that diet with 1.5 grams per megacal of inorganic phosphate had evidence of structural changes on ultrasound. FGF23, at 2 weeks of eating this diet, exceeded 300 picograms per mil in 50% of the cats that showed those structural changes and the albuminuria, and many fewer cats that remained normal. So I really just wanted to emphasize the fact that feeding diets that contain inorganic phosphate above 1 gram per megacal is potentially harmful to the kidney. And I think the study that I showed you demonstrates that feeding 1 gram per megacal of sodium tripolyphosphate in a diet that has 4 or 5 grams of total phosphate per megacal is without adverse effect. So to answer the question I was set, for azotemic cats in stage 2 and 3, I feed a phosphate-restricted diet. That means less than 1 gram per megacal, or around 1 gram per megacal of phosphate in that diet total, with N3 PUFA supplementation, and a reduced level of protein between 55 and 70 grams per megacal, but of high quality. Now, there may be benefit of different sources of protein, different fiber sources on intestinal bacteria and uremic toxin development. We need more data. But we want to ensure that the cat eats it, and that's why having multiple different types of diet out there of a similar formulation is really helpful, because if your cat doesn't eat Hill's diet or doesn't eat Royal Canin diet, then surely it will eat Purina. Monitor the response to treatment. Ionized calcium and FGF23 are really useful tools, and you can measure ionized calcium in your practice relatively cheaply. Feed more phosphate if they become hypercalcemia, and less calcium, perhaps. Maybe there are other ways of managing that, and that's an area that we hopefully will be able to address in the future. More research is needed on monitoring and managing uremic toxins and oxidative stress and uremic sarcopenia. For the non-azotemic cat, there is no evidence to date on which to base any recommendation. But that never stops us from recommending things. So I would recommend measuring FGF23 in a cat you suspect has kidney disease and phosphate restricting it, provided it's not hypercalcemic, if its FGF23 is above 400 picograms per mil. There may be benefits of other dietary modifications, but we need the evidence. I think if you're doing health screens on senior cats, try and find out what they're eating, and if their diet has more than 1 gram per megacal of inorganic phosphate, that should be a greater than sign, then advise them to change that diet. But that does require FedAF to mandate labeling of diets in terms of how much inorganic phosphate is present. And at the moment, we don't have that. But hopefully we can advocate for it in the future. I think we need something, because actually having nothing there is worse than having something that is based on incomplete evidence, but actually some evidence that is helpful to us. Thank you very much for your attention.
Yeah.
I have left 5 minutes for questions.
Yeah, we have it.
In total.
Well done. Thanks a lot for the scientific review. So there is any question from the audience? No? Okay. In the chat neither, but in this case I will do mine, which is good for me. So you said that that CKD cats usually they have dysbiosis, right? So do you think that probiotics will be something positive to supplement on CKD cats?
So that's quite early data at the moment in terms of the dysbiosis, but it certainly is a really interesting area, and how gut bacteria impact on health in general is a really interesting area. I think there are some, or there is some evidence that the different types of fiber that can be added to the diet will make a difference to the types of bacteria that are present. Whether that influences progression of kidney disease, I think remains to be seen by studies, but they are difficult to do.
Okay, thank you. Any other question from the audience? Okay, there is one question there. The mic? Oh. No? Okay, no worries. I have a solution. Yeah.
Thank you. I'm wondering if you can comment on the change over time on the blood fraction used to measure FGF23, because, correct me if I'm wrong, but I think that all the Getty studies used plasma with the Kinos kit, and now that kit has changed to serum, and IDEX uses serum. Right. I mean, there's a practical reason for that, but can you comment on, can we look at that data and the numbers and correlate it to what we're measuring now in serum?
We've certainly looked at serum versus plasma in using the Kinos assay kit in our lab, and we don't see any significant difference between those two samples. We also have looked quite extensively at storage and time at room temperature, and it seems pretty stable, actually. So the impact of transporting a sample to the laboratory is certainly not an issue for measuring FGF23, unlike PTH, which is quite labile.
Thank you.
Okay. Any other question? If not, we have arrived pretty nicely to the break on time, so perfect. Thanks again, Jonathan. Just remind, yeah, an applause. So yeah, we have a break until 4:20, and the refreshments are served in the terrace as the whole day. Thank you. See you afterwards.
Thank you, Jenny.